All right, so let's start out. Got America, my popsicle stick selectors. America, you were the first one to be selected. It's awesome. So, can you give me the location of maybe the ones on the left here? The Krebs, where does the Krebs take place? What's that? In the stroma. So stroma, that's our, and this can get confusing because we talked about photosynthesis and respiration, right? So the stroma, that's the kind of the fluid inside the chloroplast, all right? That's where the Calvin cycle happens, which is similar to the Krebs, isn't it? It kind of sounds the same. So I can see how that get mixed up. Okay, so what's the counterpart to the stroma in the mitochondria? Starts with an M. The goo inside in the middle. Let's see, I think I did put the grade on here. For Demaria, I put the, yeah, 10203.
Oh yeah.
That's good? All right. So, the stuff inside the mitochondria is the matrix. Okay? What about the link reaction? Did you get that one? No? Okay. Let's see. Willman, what do you know about where the link reaction takes place? Give me a guess. Got the matrix, got the intramembrane space. We got outside of the mitochondria. It's in the matrix as well. Matrix. The electron transport chain takes place along the inner membrane. So inner membrane cert, like right here. Yeah, that goes, the electron transport chain is right along that inner membrane. Annabelle, what about glycolysis? It's in the cytoplasm. Yeah, it's not even in the mitochondria. So it's not even on this graphic. It happens in the cytoplasm, where you have the sugar splitting into the pyruvate. Then the pyruvate comes on into the matrix. Oh, oxidative phosphorylation is this step right here where we have hydrogens going through ATP synthase. What does that look like? Where's that happening? What's this area, Julian? What would this area be? So what's this space here? See the label? Yeah. So it's between the inner membrane space and the matrix. It's along that inner membrane as well. So we'll just say inner membrane. Right along that inner membrane. The hydrogens move to the inside of the matrix. And then chemiosmosis. That happens when our hydrogens are moving across this inner membrane as well. So we'll just point to inner membrane also. Okay. Now, when this is working right, you're going to have a whole bunch of hydrogens building up in this space, a whole bunch of them. Like it says here, you're going to get high hydrogen concentration. Where are they supposed to go, Miguel? For us to make energy in our mitochondria, where do they need to go? What do you see over there? Where do they go to make ATP? The answer is easier than it may seem. What's that? Well, we're already after the Krebs cycle. We've gotten those energy-containing electron donators, NADPH and FADH2. So now we've got those hydrogens now pumped into that intermembrane space. They're all building up in that little space. It's kind of small volume, so concentration builds up quickly. In order for us to get energy, where do they need to go? Like, where do you see the ATP being made? Where do you... Oh, right over here. So what happens to make that ATP? Like, what do you see? Describe to me what you see on the screen there. To you, what does it look like?
Wait, what did you call?
You see where the ATP is? Over here?
Yeah.
So what happens to make that ATP? What do you see? What process or steps occur to drive that process?
Does it do something with the hydrogen and stuff?
Yeah, there's a lot to do with the hydrogen. What's happening to that hydrogen? Just from diffusion, molecules want to go from high to low concentration. So if you have a high concentration up here, they want to go to a lower concentration. How do they get down into here? What do they move through, Miguel? Can you read that?
The ATP synthesis?
Yeah, ATP synthase. It moves through that. And when it does, you generate ATP. Now the question is, what happens if this whole membrane is leaky and hydrogens just go back and forth? What might be the issue with that, Jasleen? What do you think? If you have them just going any which way, no order to it, how's that going to, why could that be a problem? No? Anybody have an idea? Can we tell? Well, me and Miguel, we worked out the hydrogens need to be funneled through the ATP synthase. They go through there, we're good. We got our energy. If they're leaking, are there going to be hydrogens to funnel through? No, they're not going to be as many. They're already going to go to the other side. Plus, in order to go to the other side more efficiently and quickly, there's got to be a big concentration on one side and less concentration on the other. So you're not going to have that either. You're not going to have that high concentration build up like you see on the screen. It's all about getting this high concentration of hydrogen. So if these hydrogens can just move back and forth across this membrane on their own, or maybe with the help of another molecule, you're not going to get that high buildup of hydrogen, and you're not going to get as many hydrogens moving through here. You might not get very many at all. You probably get some, but you need a lot. You know, this part generates 30 or 28 ATP per glucose molecule, so we're relying on this step to generate a lot of ATP. So if something's wrong with that membrane, Annabelle, you're going to have problems. Not going to have the proper movement of hydrogen. It's all about that hydrogen movement. from the inner membrane back into the matrix to generate that ATP. Okay. All right, so to get ready for the exam Thursday, which will be a FRQ in class, probably have you do the MCQ on your own. I want to do one, practice one in class with you right now. So everybody should have that practice FRQ. And you'll notice there's a problem with the printing. What do you notice? What issue do you notice at the bottom of that page? I realized too late.